Yeah, so first up we have this news about the B.1.1.7 variant.
Yes. So yeah, this is a pretty big news story in the UK, and I think it's. So we talked about
I think globally, I've seen,
yeah,
right, everyone's talking about it now. Mutation. It's a touchy subject.
yeah. I know our last video on it did really well,
Hmm.
which is both good news and bad news. I feel like in an ideal situation we'd just be like a boring science paper, but now people are actually genuinely interested in it. I'm like I feel guilty about we're making we're getting views off of
Well,
this.
good. We brought together stuff people can read, right? That's important.
Yeah. Um, So anyway, the first paper I want to bring up is this paper. Well, it's not even a paper. It's more like a preliminary report
Mhm.
titled "Lineage-specific growth of SARS-CoV-2 B.1.1.7 during the English national lockdown." So this paper charts the spread of the SARS-CoV-2 B.1.1.7 variant between like the 1st of September to the 22nd of December. So it's a long period of time, and during that time, the UK went into its second national lockdown. So basically, Boris was like, "We need to go into lockdown so that we can keep doing Christmas." So, uh,
Yeah, I mean that's the concern, right? Like it's where things intersect with public health, right? You know people are going to be moving around during this time. When I left for the holidays, we heard on the radio that like they did a poll. It's like people were like, despite the lockdown, people still wanted to go and see friends and family.
yeah,
So,
yeah, this is like quite interesting because like the first national lockdown was really strict, really stringent. The second one, there were certain things that were different. So schools and colleges and universities remained open. So there were certain things that you were still allowed to do. And this one kind of charts the effect that had not just on like on the whole SARS-CoV-2, but also on the prevalence of this particular strain. Um,
Hmm.
so I'm listening
And
if
we're
you can pull.
seeing this is that funny website again that came last Newsweek, right? Like not quite a journal, but it does seem it's presented very much like a journal.
Yeah. So this is where the COG UK, the group that is organizing that's been like monitoring this outbreak from the start. They've been
Mm,
putting all their preliminary stuff on this website, and
yeah,
yeah, so it's kind of an odd sort of place to put it because it doesn't have a
right.
DOI.
I mean, I think it's a really good thing. It kind of like it's like it makes it easy for these authors to publish and control comments and like see
Yeah.
responses and stuff to their work.
Yeah. So we've got a bunch of graphs, and I know that this is. I'm see if I can put this in a bit bigger, but uh, essentially like the big like the green line is the normal SARS-CoV-2 strain, and the red line is the new SARS-CoV-2 strain. And so during the national lockdown, so in the kind of grey areas, you can see the green line takes a dip under, and what the dashed line is for the R, so that's the reproductive number. So once you get that under one, then you're basically controlling the outbreak. Um,
Hmm.
so the green line seems to be controlled, but the actual red line is not controlled. The red line is still above that R1, so that means that was still spreading during the lockdowns. And
Mm.
they do some like data analysis and basically calculate this new variant has a higher R value, and that this increased transmissibility made the lockdown less effective
Sure.
because this was just so much. And yeah,
Yeah,
so
because I bet when the model of the lockdown was like initially proposed, they probably didn't have, right? They made assumptions about
yeah,
certain things, and like maybe those are a little bit different. Like it seems like we've been hearing stuff that the properties of this variant may be slightly different, right, than classic SARS-CoV-2.
yeah.
It's like, I can see that this one would spread over the other one.
Yeah. So this has got fairly sparse genomic data. So there's a lot of modeling and mathematical models just trying uh, to pick out what's going on. But they do
Mhm.
provide like some evidence for increased infectivity of the strain and kind of to justify why the current lockdown. So we're now in the third lockdown and it's being much more severe than the previous ones. Um,
Mhm.
but that's
Yeah,
so yeah.
it's good. It never hurts to have more compliance with public health, right, recommendations. And if this is the information that can convince people about that, then that's probably a good thing. I will say that this information looks really similar to when we were seeing I think I said this last week too, the D614G, right? Like we were seeing the very similar types of graphs, right? Where you see the prevalence of a variant
Yeah.
kind of take over, right? The existing ones.
I
Uh,
feel like D614G didn't have such a drastic change in the R0. I think most of them would seem to be like competing with other strains so much. So
yeah,
it wouldn't so with those it wouldn't be so much that the lines are different. The lines would be like almost add up to the same thing, but the way they'd wave. Whereas like with these two lines, if you kind of add them up, they'd be bigger.
yeah.
Whereas with
Sorry, I don't mean the R0 graph. I mean the top graph, the one
oh
that has just like new cases.
yeah
Right? Like those in that new cases graph, right? You can see it's between the red and the green,
mm,
right? So like the red variant is above now the other lineages,
yeah
right? Indicating that it is the more prevalent version, right? Like
yeah
something's happened in the last it's pretty small window of time, right? In the last two
yeah
weeks or something, at the beginning of December, where now this is gaining prominence. So that would be the indication. That's probably the types of differences people are looking at when they're making this call and saying it's more infective, it's more
yeah
transmissible.
yeah. And I think that's going to bring us to the second paper, which is from an external group that also did some work trying to estimate the transmissibility and severity of the SARS-CoV, estimated transmissibility and severity of novel SARS-CoV-2 variant of concern
Yeah,
2012 numbers 2020 12/01 in England. So this is again, it's quite clearly not peer reviewed. It's still, but they so this is while the previous paper came directly from the COG group, this it was very preliminary. Well, this one is like a bit more filled with stuff. So they got like they're looking at the same data, but they also combine that with mobile phone data to estimate the
hmm
number of contacts, and then put that into the modeling to see how effective that. Because there's always that theory that maybe it wasn't the strain's more transmissible, maybe it's that the lockdown failed. Um,
yes.
so this the aim of this is almost to try and
Yeah,
like
to
analyze
really, I
that.
mean, yeah, because again, we only have this one window, right? But before we can do any animal testing, we only have the window of this epidemiological surveillance that we're up to.
Yeah.
And so, of course, you got to do the right controls because that data is not is not gathered for the purposes of determining this information, right? You have to remove other possibilities.
Yeah, and I think that this was published on. Let me double check this. Yeah, this was published on December 26th. So, to put that into context, this strain was announced on December the 19th. So,
Mhm.
this is equating to about a week's worth of like work that
Yeah,
had to be put in very rushed. An animal study requires like, depending on what you're doing, you need to order animals, you need to breed them, you need to make sure. So animal studies might take
absolutely.
about a
Yeah,
month or two.
won't have those yeah, and we said this last news week as well, right? Like for people who make decisions, right, in policy and so forth, right? You kind of need faster, you have to make your decisions on like limited amounts of information. This is the challenge, right, of navigating our society using science, and like it's a good way, right? Because we can verify and we can like go back and say what did we do right and wrong based on how we acted, but we do have to sort of like, just take the little hints that we get and then make a plan from that.
Yeah, I mean, this actually does a lot of that planning, and they don't just monitor the outbreak so far. They also do make some predictions. So they
Mm,
predict. They try to analyze like what kind of um, what kind of strategies to managing the outbreak we should take in future, and kind of predict how ICUs will be overwhelmed based on this new strain and this new R number.
sure.
Because um,
Yep.
while this variant
Yeah.
isn't,
Updating the models with this information
yeah,
and then yeah, regenerating yeah, a set of best practices guidelines.
yeah, because I mean, people will be getting it faster, so ICUs will be more likely to be overwhelmed faster. Also, the herd immunity threshold may have to be much higher. So you have to put that into calculations of how many people need to be vaccinated, how many people need to be put in isolation. So this is quite
Yeah,
an interesting paper from that decision-making perspective of like putting yourself in the
sure.
position of the policymakers looking at this, and the information isn't perfect, but you still have to make a decision on it, and that's a lot
Yeah.
of
Yes. That's the scenario we've been navigating probably for this entire pandemic.
yeah. I mean, waiting and seeing often is until all the data's in can create a massive delay that means all your things you can do are kind of less effective.
Yes, exactly. The boat has sailed on those things, right? Like
Yeah,
we're okay. Well, up next we have
S
S
gene.
gene dropout patterns in SARS-CoV-2 tests suggest spread of the H69 deletion V70 deletion mutation in the US. So
Yeah,
this is like early surveillance in the US now of the same one of the loci that is involved with the B.1.1.7 variant.
yeah, I found this interesting. It comes from a genomics testing company. It comes from Helix, and so they've been doing some of the COVID testing in the US. And
Mm-hmm.
and one thing is that is known about this new strain is that the H69V70 deletion can stop the kind of primers that bind to that gene from working. So the specific test for the S protein stops working sometimes if you've got that deletion.
Gotcha.
So what they've been doing is they've been looking at other genes as well. So throughout the outbreak, they haven't just been looking at the S gene. They've also been looking at the N and ORF1AB genes. So they've
Hmm.
been, and so based on that, since like this outbreak is going on, they've been doing this like test for three like different genes, and they've noticed that there is an increase in the dropouts of.
Yeah, for this one this one loci that they test. Yeah, I mean this is sort of a great callback to our discussion from last week, right? When we were talking about coverage, right, over the genome and testing. Yeah, very exciting stuff, like in terms of just letting people know that, not all that you're gonna have to cover more bases as the virus changes, right, for these diagnostics.
Yeah, the interesting thing is like so when they heard about this new strain, they went back over their records and to see the number of tests that register this kind of dropout, and they found there was like a spike in October, which kind of
Hmm.
rose to in November in various states. And
Hmm.
well, they do kind of clarify that this doesn't necessarily mean that new strain is in the US, because obviously this deletion mutant has evolved multiple times independently.
Yes,
But
that's right.
so it could be a new strain, but the timing is the key because that peak in November also kind of correlates to when the B.1.1.7 strain arose. So it does
Hmm.
seem to be like kind of more than a little coincidental that these both appear at the same time. So uh,
Yeah.
it
I
could.
mean, I would say this, right? Like, right, mutation and selecting for, this terrible experiment of like of human people, right? Selecting for those mutations, it requires like a lot of people to have infection, right? So if your country has a really high amount of infected individuals, there's more of a chance, right, of seeing mutations. And if those mutations are beneficial, as we're seeing some evidence that like at least when put together, they're beneficial in the B.1.1.7 variant, then they'll spread in the population, right? And
Yeah,
then maybe they'll until you kind of settle on the successful variant, and that one takes over.
yeah. Um, so yeah, I mean they've got this map of the US on as Figure 2 where they show like the proportions of times that that strain appears, and
Mhm.
so there are peaks in certain areas, and the one thing I'd like want to know is like whether how systematic their coverage of testing is because there might be some areas where they don't do much testing at all, so they wouldn't know. Um,
Yeah.
I mean, the US is a giant country with lots of different like states that have their own systems. So, as an outsider, I find it very difficult to navigate this sort of thing. Um,
Yeah, I mean, the other thing that I'll say is complicated about this is just from their company's data, right?
yeah, exactly.
So it's like, so hotspots also appear proportional to how much they're collecting in those areas.
Yeah,
Yeah, because there's like a patchwork of companies that would do testing probably.
mm, yeah. I mean, because one company isn't necessarily big enough to do the entire US. Uh,
Hmm. Hmm. Which again, I mean, I don't want to throw so much shade on the American healthcare system, but like, that is a weakness, right, of non-public healthcare systems, is that sometimes it may be harder to, unless there are open standards that allow
mm.
that transmission of that information, it may be harder for someone to do the single analysis and gain like a trend across the entire
I
country.
mean, it's also like a massive country just in terms of numbers. So there's a
Oh,
lot of
absolutely,
infrastructure that needs to be put in.
absolutely.
So I think a lot of stuff does need to be handled statewide. Um,
Yeah. Yeah.
But so like I'm going to be flipping off to another papers because
Yep.
the
Let's.
UK has a similar sort of system. So this is from a UK test and trace lab that has also been monitoring dropouts. Um,
Mhm.
so like the UK, much smaller country, so again we don't have to deal with the. So this is very much a nationalised sort of event. So it's part of Public Health in England.
Mhm.
So it comes from a government-related laboratory. Um, and it investigates the prevalence of gene dropout. But they ask a slightly different question. They look at this thing called the cycle threshold value for RT-PCR. So that's basically the time it takes for. So we talked about this a little bit last week. The time it takes for the test, the the PCR test to reach saturation. So
Yes.
if
Yes.
if you've got
And so
more,
like the sooner it happens, the more sample we started with, probably. Right? Meaning a
right,
higher amount. This is the quantitative. This is how RT-PCR can be quantitative, right? Because the faster something amplifies to that threshold, right, it means that there was more of it to begin with.
yeah. So they measure the other two genes and see how. So they did the similar kind of tests, but they measure like how quickly those other two genes meet the cycle threshold, and what they
Mm.
find is that in general dropouts tend to reach that threshold much faster. So meaning that they've got more genomes.
Yeah.
So
Yeah. So this is nice. I think this is a nice complementary one also to this idea that this could be a more successful variant, right?
yeah, um,
Yeah, I guess sample collection is like one of the things that I would wonder about this one. Like because it could also be something like what are the criteria in which they admitted samples into this? If it's totally unknown, right,
yeah,
then you don't know. More people are getting. Maybe it makes something about this infection makes people want to get tested sooner. It's probably Sentry though, right? This is just um, like an overview, like a sampling of everybody in the country.
yeah. So this is, I think,
Mhm.
a sampling of people. I
Yeah.
mean that, and yeah, I mean it's not like they're dealing with equal groups, so it's not necessary. And the actual like kind of difference is relatively subtle.
Mhm.
So it's so
Mhm.
wouldn't say it,
It's
and
also, yeah, it's from the same. I also think it's like fundamentally the same data, or no, it's a different analysis, right? Like
mm,
they're trying to correlate. Yeah, the CT is a different readout of success, we'll
mm,
say, right? Success of the virus. So that's good. I do think that this adds like a little minor, right, bump to the idea. And again, taken all together, this is what people are making decisions off of, right? So,
yeah, and I mean
yeah.
it could add another piece of the puzzle of why this strain is more transmissible. Uh,
Hmm.
I mean, I guess I probably should disclose that number of genomes is viral load, so that doesn't necessarily translate 100% to viral titer, but it's still very much an indicative sort of thing to think about.
Yeah.
Uh, and the next is quite an interesting paper. It comes from Pfizer.
Hmm.
So, neutralization of N501Y mutant SARS-CoV-2 by BNT162B2 vaccine-elicited sera. So, will this strain be neutralized by vaccine sera? So
Uh-huh.
if you're immunized with the BioNTech vaccine, are you going to be safe? Um, so they
Of
answer
course,
this.
they're putting this one out because
Yeah,
they want to make sure people are excited to use their vaccine.
yeah. I'm surprised none of the other vaccine companies aren't just rushing out stuff because this is like a 3 page long paper. It's very short, but they just
Hmm.
like pull out the kind of they've got this PRNT50 data where they pull out and they kind of say that okay, there's not much difference between the old Y N501 value and the Y501 value.
Yeah.
Um,
I think it's good. I mean, this is the type of stuff. Yeah, I agree. Like, to allay fears, people for different vaccines, it would be nice to see. But I also think seeing it for one vaccine also sort of makes you think that it would be okay in the other
mm,
vaccine scenarios.
yeah,
Um, yeah, just because like you're getting the same spike protein mostly in each one, right?
mm,
And like the idea of this mutation is this one sits in the receptor binding domain. That's like the oh man, like that zone could be one of the zones that like
yeah,
doesn't recognize. But yeah, it seems I guess this still works for the neutralization. It still neutralizes, right? That's what it's saying.
yeah.
Yeah,
It
still.
still basically neutralizes. I mean, for their sample size, they didn't really see much of a difference in the variation.
Mhm.
So, um,
But again, I don't think that's the primary. I don't think when I heard this news about the variant, that wasn't the primary concern in my mind
mm,
about the variant, right? Like, oh yeah, I believe the vaccines will all work against it actually. Like just from like my intuition about the science, right? Of course, it's good to see that data like this, but
mm,
my concern is more that transmissibility or like that it goes to higher titers in people, right,
yeah,
quicker. Because that just means that it's more difficult to control, right, on the big scale, right? Because I do think that that's the biggest problem, the big problems we've been having. I mean, even the variant itself is a product, right, of the fact that so many people are infected, right? If there were fewer people, we wouldn't see the emergence of variants like this, right? Because there wouldn't be that large population to play evolution in. Um, so yeah.
Yeah, I mean, one last thing. They sorry. They look at just the N501Y mutation, but the
Mhm.
new mutant has a bunch of other mutations in it that could.
Yeah.
So the H69 B70 deletion, like so, last time we talked about it, we had this paper where the N501 mutation like kind of was theorized to increasing by about 20%, increasing transmissibility, and then when the H69 B70 came in, that was a much bigger increase.
Yeah, another jump.
I'd be very interested to see what that effect would have on antibody responses, or just trying to figure out what the actual fuck is happening with the spike protein when that happens.
Yeah, for sure. What's the I mean, like, yeah, in some ways, like the golden piece of information that could help us answer some of those things is like, yeah, this modified spike, because it's all these modifications to spike protein, right? What does it, how does that change its behavior in its interactions that are biologically relevant, right?
Mm.
With receptor. Yeah. And again, those are stuff that probably the experiments are being done now, right? Like
Yeah,
that's the nature of science. Like we're not the first people to think about these things, right? We're talking about them, but like inside of labs, people are thinking about these things, setting up those experiments, but it takes time.
yeah,
And so you have early decisions to try to get ahead of potential problems, and then yeah, and then we have to wait to see some of those confirmations. Well, and nerd out about them and say
hmm,
like what we think is like happening, right? Like try to guess, yeah, exactly what's going on at the molecular level.
yeah.